Hello, everyone. My name is Liana Shvachiy. I'm a PhD student in biomedical sciences, and I am here to welcome you all for the Female Clinician Engineer webinar series. Today is the second day of our great international female engineers that will present their success stories. So for today, we have two amazing speakers. Unfortunately, we had a last-minute cancel of our first speaker that will be presenting on the 25th of July. So first of all, we have some ground rules. As you have noticed, the sessions will be recorded. Please do not turn your camera and microphone during the sessions, but you can always ask questions in the chat. We are always welcoming questions, and the more the better. And of course, stay with us until the end for a nice group picture. But most of all, please enjoy, learn, and have fun. So I am happy to present our first speaker, Professor Dr. Marcy Zenobi-Wong, who is the head of Institute of Biomechanics, and she will present her work about biomaterials for treating skeletal diseases and injury. Please welcome Professor Marcy Zenobi-Wong. The stage is yours.
Thank you. Can you see my screen? So, hello everybody. It's a great pleasure to be part of this webinar, and I would first like to thank Liana and Banu for inviting me and for organizing such a great initiative in these challenging times. I think these webinars are really one of the positives about this Corona time. I just also like to say a few words about who I am. Maybe you can tell from my accent that I'm an American. I did all of my education in the US, East Coast, West Coast, and also in the middle. And at graduate school, I met my husband, who happens to be European. Well, actually not European, he's Swiss. We got married, and he convinced me to try out Switzerland for 2 years, and that was more than 20 years ago. So my whole professional development has been in Switzerland. Which has been very interesting, and I'm happy to entertain questions at the end also about not just my research, but about my career path, which was quite different because I was the trailing spouse for many, many, many years, and until I finally got my professorship at the age, I was an assistant professor at the age of 48, which I think was probably the oldest in Europe. But anyways, what I would like to tell you, talk to you today about is the work we do in my laboratory at ETH in Zurich about biomaterials. And trying to use biomaterials in new ways to treat problems of the skeleton. My background is mechanical engineering, but I have, over the years during my PhD, my postdoc, and then position at senior scientist, and then assistant professor, I've been getting more and more involved in biology and material science. And what I find truly fascinating about this field is the ability to combine these different fields to try to come up with new solutions. So I think this is a great lineup that you girls have put together. So thank you very much. The other thing that is really important for me to say at the beginning is that none of the work that I'm going to present is done by my own hands. It was done by my wonderful team of PhD students. This is a picture from 2016, and they've all, I think, with one exception, have finished their PhDs by now, but they have actually done the work that I'm going to be talking to you about. So just thanks to them. My laboratory is particularly interested in the tissue cartilage. So cartilage is the thin layer of tissue which lines diarthrodial joints and allows the skeleton to move. And the lab was funded actually for many years by FIFA, the football organization, because they were very interested in coming up with therapies to prevent or to help the professional players recover from injuries. It's a huge, the cartilage degrades, unfortunately, and it causes huge socioeconomic costs to all countries. And I just have some statistics. These are US statistics, and it's really true worldwide that it is one of the leading causes of disability in the US, but in almost every country. And this is because when cartilage degrades, it's a disease called arthritis, which I'm sure everyone has heard about. And it's a disease which will cause disability, but will never kill you. And what ends up happening is that it generates decades and decades of emotional and economic costs. So just a few of the statistics. There are 52 million adults that have been clinically diagnosed with arthritis between the period of 2010 and 2012. It has cost over $100 billion in costs per year. But the real cost, I think, is in the limitation of movement of the people that suffer from this disease, because it's painful to move. So they have problems, they're limited in their daily life, often cannot work, have trouble climbing stairs, can't go for a short walk. And in summary, these are millions of people that are denied the benefits of exercise. And we know how much exercise has cognitive and physical benefits. So with arthritis, you don't just have problems in your joint. There are so many comorbidities like depression, like cardiovascular disease. And it's a huge problem to society. We know quite a bit about what causes arthritis. So there are risk factors. If you're female, if you're postmenopausal, if you're obese, some genetic factors, but if you've had a previous injury, like you've torn your ACL, these are all going to increase your risk factors for getting this disease. And the bad news is that despite many efforts on the part of pharma, there are no disease-modifying drugs to treat arthritis. I'm talking about osteoarthritis in particular. And the only thing that the medical industry has to offer these patients is a total knee or hip arthroplasty, meaning your joint will be cut out and replaced by something artificial. This is a successful surgery for sure, but the problem is people are getting osteoarthritis at a younger and younger age, and it becomes problematic when you need to put an artificial joint into someone who's young and active, because these joints have really been designed for elderly, sedentary people. Yeah, so this is a big problem, and there are so many research groups working on topics to try to stop the progression of this disease. And I just would like to show you a few examples from my research group where we're using particularly biomaterials in one way or another to try to understand if we could contribute to a solution for this disease. And I have to say we're far away. Most of the work that we do are really still at the basic science level. Yeah, so if you were to look inside a knee joint, there's a kind of a cartoon shown on the left. On the left side of this cartoon is what a healthy cartilage would look like, a healthy joint would look like. And you can see this layer of tissue, which is smooth. And the joint itself is filled with a fluid. It's quite viscous, like honey, a synovial fluid. And this really helps the two surfaces to glide across each other, which allows our skeleton to move in such an elegant way. But we have, together with a former postdoc from me, Gonzalo Barreto, have been looking more at what happens on the right side of the figure. And there's a lot of kind of bad stuff going on if you have the beginning of this disease. Often the disease starts with a mechanical insult, which may disrupt the tissues of the joint. And this will cause direct injury, but will also start the cascades of inflammation in certain cases where the material, the living material, when it's injured, will then start to release fragments of the cartilage tissue, for instance, like fragments of collagen or other molecules. And it's been acknowledged that many of these fragments that are released are actually inflammatory itself. And so in this disease, you kind of have a downward spiral of your tissue being injured, and then there being release of fragments. This is causing inflammation, which is then causing more degradation of the surrounding tissue. And what you have often over decades really is this kind of downward spiral of the tissue getting softer and softer and being chronically inflamed. And so the question for us was, when one has a situation like this, at what stage could one intervene with a biomaterial? And so I just wanted to show you very briefly 4 different examples of where we've used biomaterials to try to propose a solution for this problem of cartilage degeneration. The first one is a fun project that was initiated by a colleague of mine, Eddie Benetti, who's a polymer chemist, and we were sitting and thinking, wouldn't it be nice if one could come up with a polymeric paint to kind of coat the surface of the degraded cartilage and restore the lubrication, the lubricity of the surface before one, sort of to stop this downward spiral. We have also had studies where we're trying to deliver certain drugs to cartilage. And because of how dense cartilage is and how it's negatively charged, it's been really kind of tricky to get drugs to go to where they're needed. So I would like to show an example of that one. And then an example of if you have an injury and you want to prevent it from kind of getting bigger and bigger and eventually causing arthritis. This is an idea we had, that we could take a material that is injectable and kind of use it to fill the cracks and prevent this progression of disease. And then finally, just have a few slides to show how we're also using biomaterials in a 3D printer to try to make an engineered tissue more similar to what you would find in native cartilage. So those are the 4 things that I'd like to briefly touch upon today. Yeah, so the first one is this cartilage paint project. So as I mentioned, the friction between 2 pieces of cartilage is like ice on ice. It's almost zero. The surfaces are so slippery. If you try to pick up pieces of cartilage, it's really difficult because they really slide. So the tissue has been evolved to be like to have almost no friction as it moves. And there are many people that have studied the physics of how this low lubrication starts. And it's been found, well, it's a complicated situation, but it's been found that there's a molecule that is expressed by the cells right at the surface. This molecule is called lubricin, and it binds in a non-covalent manner to the surface of cartilage. And because it has this brush-like hydrated middle part of its domain, it becomes or renders the surface very slippery. So what you see here is just a cartoonist imagination of how lubricant would be attaching to this cartilage surface. It binds and unbinds. You can shear it away and it reforms. And if you have no lubricant, so this can be in a mouse model, it can be knocked out, you find that these animals actually get arthritis at a much higher rate. So it really plays a role in a surface being slippery. And so we're interested in what causes the cells to start expressing this molecule, and actually the shear stress is one of the factors which causes the cells to express it. But once you have lost this surface layer, once you have the early stages of arthritis, you have lost this low friction environment. The first thing that happens when you have the disease is the very surface of cartilage begins to show fibrillations, and the surface becomes kind of rougher. And it was at this point where we thought, could we have a synthetic material that kind of would play the role of lubricant, that we could just incubate on the top of this surface to kind of rescue it from progression of disease. And so this is a work from Eddie and my very talented PhD student Julia Morghese, and this is the solution that she came up with, and it's got 3 parts. It's not important to understand the chemistry so much, but to just realize that it was designed to stick specifically to the areas of cartilage where you had this fibrillation and the proteins were exposed. And so the proteins have this functional group, this amine group, and it was only in those places where the cartilage paint could covalently attach. So the cartilage paint had this black bone. This is the black wiggly line. And Julia attached groups that would allow it to covalently attach to the proteins, the exposed proteins of the cartilage. But she also decorated this backbone with these red brush-like molecules. This is a synthetic polymer that is one of the polymers that is the most hydrophilic. So once you have this paint applied, these brushes kind of form this lubricious environment on the cartilage surface. And so I wanted to show you some in vitro results that Julia obtained, looking exactly at the coefficient of friction of different surfaces. So the machine on the right is measuring this coefficient of friction. And what Julia did is to measure the coefficient of native cartilage. This is this NC. And you can see the values are between 0.04 and 0.08. So it's a very low slippery surface, as it should be. She then tried to simulate this disease state by just digesting the surface. She took healthy cartilage and digested the surface and found that the coefficient of friction then doubled. And this makes sense because it's very, it's lost this smooth surface because it's been digested and the surface is now a bit fibrillated. She then incubated her cartilage paint onto the surfaces and found that in most cases, that the coefficient of friction with different grafting densities of these brushes was even lower than native articular cartilage. So they became really, really slippery. And so this was really exciting to us, and we are now taking this polymer and we're now trying to treat animal models of arthritis to see if we can also prevent the progression of this disease. Yeah, so that's one thing she found. She also, being such a talented polymer chemist, could change the the shape of these polymer brushes. So in this study, what Julia did is she took a native cartilage surface and then exposed that surface to some kind of degradation. And this was using this enzyme you see here called chondroitinase. And to make a long story short, this enzyme works on the surface and starts degrading some of the molecules within the cartilage. This is usually stained by red, so you can see that the red on the top of this picture doesn't have any red anymore. And she could then expose it even more, and you can see the red has gone lower. But she then asked if she then put her cartilage paint onto this surface, could she prevent this effect of the chondroitinase enzyme? And the result is interesting because, as you can see in the schematics, Julia was comparing if you have a linear conformation of her polymer brush, so they're all sticking up like this, or if you compare it to a conformation where the polymers are actually cyclic. And the cyclic conformation of these polymers actually was better at preventing the degradation of the material. So this is just an example of how the structure of a polymer can actually have a function that could be relevant for trying to protect these cartilage surfaces. Yeah, so I'd like to come to my second example. This was a project done by Florian Formica, and he was interested in a drug, a very common drug called dexamethasone. You might be hearing a lot about it recently because it's been found to be effective by some people to treat COVID disease. It's a glucose steroid, and it has strong anti-inflammatory properties. And this would be very nice to deliver to an inflamed joint. The problem is if you take this drug, dexamethasone, and just inject it into the joint, because there's so much turnover in the joint space, so the fluid gets rapidly turned over. Every time you inject a drug into the joint space, it is rapidly cleared. So it's very difficult to target cartilage through an injection. But what many people have realized is that you can use the charge of cartilage to make more effective drugs. Cartilage is a tissue that, because of its biochemical composition, has a very high negative fixed charge density. And if you can link your drug to a positively charged entity, you can get it to rapidly target the cartilage, and the cartilage then binds the drug due to this electrostatic interaction. And so what Florian did is he conjugated this drug dexamethasone to a positively charged polymer. In this case, it's a polymer called chitosan, and found that you can really get the material to stick to cartilage, to penetrate cartilage, and be retained there. So in a sense, what you're doing is to render the diseased tissue itself to be like a reservoir for your drug. And this is just shown by this green staining. This is a fluorophore called fluorescein, where it sticks. If you've got this conjugation of the drug to a positively charged entity, and that if you don't have this positive charge, you lose, the drug will be in and then be washed away. So we're interested also in pursuing this prodrug in the context, the next step would be in the context of an animal model of arthritis. Okay, so the third example that I wanted to share with you was an idea that was motivated by the fact that many people that play sports at the elite level, because of the high impacts they subject their skeleton to, a lot of them have cracks in their cartilage. And it would be a shame to let these cracks progress and then eventually involve the entire joint in osteoarthritis. And so we had the idea that one could develop a material that one could inject into these little micro cracks. And injection is good because then it's a minimally invasive application of the material. You don't have to have a big surgery. Three students of mine, this is Nicolas Brogier in the middle, Emma Cavalli on the left, and Clara Levinson on the right, we're working quite hard to develop a material that you could use to inject, because this material would have to have certain properties. It would have to be cell-friendly. It would have to be compatible with what the cells of the joint want to do. So they want to divide, they want to secrete extracellular matrix. And the important quality that this material needs to have is that as it's injected and begins to polymerize in the joint space, it's very important that it would also adhere to the joint space. So it needs to also have this adhesive property, so that the repair tissue is actually integrated into the joint and not just floating around where it wouldn't be functional. So these students worked quite hard to come up with a material that would fulfill all these different requirements. And this is a schematic of the material. It's based on a carbohydrate, which is found naturally in the cartilage extracellular matrix, called hyaluronin. And as was described in these publications, they did was to modify this carbohydrate chain with two peptides that would allow this material to form a hydrogel in the presence of an enzyme. And they were able to achieve the adhesive properties because these amino acids that are substrates for the enzyme also are present in cartilage tissue. And on the upper right, you can see that the gel is actually able to stick two pieces of cartilage together. And if you remember, cartilage is very hard to get things to stick to cartilage. So it has the adhesive properties. And also, if you look at the picture on the lower right, you can see this is a histological section of cells from cartilage that have been dividing in this material. And the brown stain is a stain for the prototypic collagen type that they are supposed to secrete when they're healthy. So it's a material that, has been tested in large animal models now, and we're hoping could play a role in prevention of diseases like arthritis. And then I quickly come to my last example, where we have recently gotten interested in seeing if we can 3D print some of our materials. And this is the idea behind this, is that if a patient has a particular defect from a sports injury, for instance, that defect, that geometry of that defect is really specific to that patient. And wouldn't it be nice to be able to manufacture a living graft that suits exactly that patient's injury? And so we've been working for several years to try to develop materials that one could put into a 3D printer. And the idea would be to have perhaps multiple different cartridges where one can print in a layer by layer fashion the different layers of cartilage in the presence of cells. And so this is some of the guys from my lab that have been developing the bioprinting, and you can see it's the type of printing that we're working with is extrusion, and kind of works like the model being shown in this video. Yeah, as it turns out, the big challenge in bioprinting, at least cartilage, was to get the flow properties of the materials that we're printing to be right. So that you can see in this video is a material, we call it a bioink. And this material's rheological properties have been kind of engineered so that the material can flow through the nozzle during the printing process, but then stops the flowing after the printing has stopped. So you can see this nose structure. It's a paste. It hasn't been cross-linked yet, but it's keeping its shape. It's not that you print and then the fidelity of the print becomes less and less. And so this actually turned out to be a material science, material flow problem that we were working on. And since we've gone through and tried to develop these bioprinting materials that also fulfill multiple functions. This is the tricky thing about working in the body, is it has to be cell compatible in the synthesis and in the cross-linking, but also has to be, treating the cells really well and giving them all the signals and morphogens that they expect. So this is, I guess, summarizes well the challenges that we're facing in the lab. Yeah, and with that, again, acknowledgements to the talented students that have been working in my lab and that I've had a privilege to work with. So I think I can stop there, and I'm happy to take questions if there are any.
Thank you very much, Professor Marcy. We have some questions, mostly from me and Banu. So Banu says that you have a workspace that says that have a collaboration between highly complex areas like self-mechanics, biofabrication, advanced materials. Besides producing absolutely satisfactory results, what are the difficulties what issues require patience and hard effort?
Yeah, so I think the biggest struggle it's very easy, not easy, but it's really doable for a research group to make a study with a new material, to test it and get a publication. This, I think, there's thousands of examples of this. I find the regulatory environment to translate biomaterials is challenging and is becoming increasingly challenging with the implant file scandals of late. And we feel really strongly that in rare exceptions, not only do you need a biomaterial, but you also need cells. And in the best case, you have cells from that patient. And this really, ups the complexity, regulatory complexity of these kinds of products, orders of magnitude. And it becomes a situation where it's no longer the domain of the academic group. And so I think, at least at my university, they're strongly supporting transfer of technology to spin-offs or to companies, just because I think it needs to be very clear, what is the domain of the academic and where it stops. And doing these kinds of studies, even the preclinical studies that need to be done so that they're acceptable to agencies, I'm not sure I'm the right person to do this. So I find that transition is going to be challenging.
Okay, thank you very much. Actually, I have the same opinion that the preclinical studies are very important, for sure. and we need to translate them, but it's always a challenge for us to do that. So we have to be very careful. And of course, we have to lead it to the other experts in the field to do it. We have another question. So you have works where you use 3D printing technology. And as a member of a women in 3D printing community, what do we need in additive manufacturing field for developing cell-based biomaterials, hydrogels, and scaffolds? What should we expect in new age bioprinters?
Oh, that's a great question. I wish I knew the answer. Yeah, so we've recently wrote a review on this topic, and there's a lot of development. There are basically two kinds of printers, if you want to generalize it. One of them is forcing the biomaterial and the cells through a small orifice, and this is what gives you the resolution. So we call these deposition-based techniques, and so the printing that I showed, everything is going through a nozzle, and this is what gives you the resolution that you have. The effect of the cells on this extrusion process is unknown, and I think needs to be addressed. Like, how are you actually affecting cells when they're printed? There is a way to avoid this, and this takes you to the second kind of class of bioprinting. And here you don't, you start with a vat of liquid precursor, and then the structure that you want to print is actually polymerized from within this bath. The problem with that type of printing is that you need to, this is light-based, so you need to modify your polymer so that it can be cross-linked with light. There's usually an involvement of a photoinitiator. Often the light is UV. So you don't have the problem of shear stress on the cells, but you have the potential problems of I don't know what the UV light photoinitiator could be doing, but both of these I think make translation difficult. So each printing method has its advantage and disadvantage, and there is not one that is working, I think, perfectly for all applications. Yeah, so to make a long story short, I think we don't have the solution yet, and we don't have the material. It would be great if there was a naturally occurring photosensitive material that was cell-friendly. That doesn't exist yet. So I think there's going to need to be progress on many fronts before we'll have clinical products.
Okay, thank you very much. So another question. We just saw how wonderful of a scientist you are, but we also know that you are a mother of 4 children. So how do you take the best of both worlds, and how can you harmonize all that and work in both fields, let's say?
Yeah, I mean, my children, my oldest daughter is 13 now, so I feel like I've gotten over the most difficult spots. It was not easy. I was working, part-time for many years. I didn't have a permanent position. I was in Switzerland because of my husband's job. There were, challenging times. And if I were to give some advice, is that I never, even if it was really challenging, I never stopped completely. I always tried to have even a small project running, or try to go to a local conference. And I think this was an important thing. The other thing, and it may have something to do with being in Switzerland and not having my family and my support nearby, is that in many respects, my job saved me. So it was my real connection, and it was also challenging, but also a good balance. So when you know, nothing worked in the lab and papers were rejected. I feel like at least I have my kids. And then when, you know, the kids are just, you know, you just need to get out of the house. I was super glad to have my work. So it was hard, but it was in a sense nice to have both of them, especially being in Switzerland. But yeah, I would say just try to keep some kind of contact to the research.
Thank you. Yes, science can be frustrating for some times, and we really have to keep on going. So what was your motivation to follow this area in specific, this research area?
Yeah, so I've been from my PhD always a cartilage person. It sounds like a really simple tissue compared to many tissues like the heart or the liver. It just has a single cell type, and it seems very simple, but it's not. There's still so many things that we don't know about this simple tissue. So this is what I was studying with my PhD, and I'm still fascinated by it. The cells are really special. They have properties that you don't find in any other cell types. Like they like low oxygen, and they're really fascinating. And then yeah, it was, I think, when I started my position here, just being in contact with people that were interested in biomaterials. Sometimes they have a biomaterial and they didn't know what application. So it was kind of to have you know, looking for people that were interested in cartilage and might have something that some material that could solve some of the problems.
Okay, thank you. So have you done any of the human testing in human population, or you only did the in vitro and in vivo studies? And what kind of animal models do you use?
Yeah. No, we have not started clinical trials, and again, I don't think this is something that is going to be possible in my lab. We do in vitro, and then usually the sequence is we do lots of in vitro characterizations, and then we go to an in vivo model. Interestingly, we found in many cases that the in vitro does not predict what we see in vivo, which kind of makes me question if we should be spending so much time on in vitro results. But the models vary. If we're just looking at biocompatibility, this will sometimes be just a implantation subcutaneously. The biggest, most realistic animal model is done in sheep, but this is also, you know, comes with a lot of disadvantages, particularly because they're so expensive, especially in Switzerland, that you don't have very many chances to try. You basically have one shot, and this is yeah.
It is challenging.
Challenging, yeah.
I can tell that because I also use in vitro and in vivo models, and it's challenging to go from in vitro to in vivo.
Yes, it is.
So we have some more questions for you. Can we say this, as the implant was grown from the cells, the risk of rejection is far lower than for an implant made?
I think in general, if it's an autologous cell, the cell itself will not be a safety problem. I think this is clear. The reason that we are so fixated on this hyaluronan backbone or starting material is because it also is a natural molecule that is found in the joint. So I think what we're aiming for is a solution where the scaffold that you provide the cells is so cell-friendly that the cells are encouraged to right from the very start secrete a lot of their matrix. So the cells themselves are kind of infiltrating this material, and the assumption is that when it's implanted, the immune system is just seeing these proteins and these cells, and the material is really secondary. It can be very different though if you use a scaffold, which because of whatever property is causing an immune response, even if you have autologous cells.
Okay, thank you. So are there any patents that you received in your work that turns your study into a product? Can you tell us about them, if you have them, if it's difficult to get them?
Yeah, so this is something that we think about a lot, particularly because I think we have an obligation actually to explore the potential IP of what we do. And so we have a process in the lab where, before something is published, we have a discussion to see if it's protectable. There's a possibility at ETH where you can go to the patent office and have a search done. And we will, often if there is something to protect, we will also do this. I think it's an obligation.
Okay. So you have a study area that combines many fields such as biochemistry, biophysics, mechanics, material science. This shows that it is a result of a very harmonious teamwork. What do you look for the most when you are working together? How do you choose your PhD students or postdocs?
Yeah, that's a great question. I don't have a secret formula. What I like when I hire somebody is that that person knows how to do something that I don't know how to do, or our team doesn't know how to do. So if someone tells me I can make a hydrogel with chondrocytes, I'm like, oh, well, we already know how to do that. What else can you do? And the second thing that I really like is this interdisciplinarity. So I don't want to have 90% chemists. I really think it's important to have a mix of biologists and engineers from all the different disciplines who are willing to make the effort to talk to each other. So the social competence is also really important. So when a person interviews for a position, this person needs to get along and be willing to talk to people from other disciplines. And some people are just really naturally curious and really want to do that, and some people are more focused, and that's also fine. But for my group, it's much better that you're open to other disciplines.
Okay, so we have a comment and a question together from Miriam Smith. She refers to the Banu's question about the patents. She was told during her tissue engineering course that it is not possible or very difficult to patent discoveries in tissue engineering, as cells can be patented. Could you elaborate on this, if this is still the case, or if so, how do you deal with this situation when you try to valorize your discoveries or your research?
Yeah. It's difficult to get a granted patent for sure, because as you know, these patents are written so broadly and cover, I don't know if that's a good thing, but that's basically how it is. But I think there are things that we have patented do not involve cells, but will often be a processing technique of a type of material. And I think, yeah.
And have you thought about a startup within your lab?
Yeah, there's been two. And ETH has a program which is called the Pioneer Fellowships. And it's exactly to help people that have discovered something in their PhD or master's thesis. It gives them, I think, 18 months of funding where they can try. So
that's very exciting,
yeah.
because entrepreneurship and innovation right now is very important, especially in research. So we have to go beyond our research. So thank you very much, Professor Marcy Zenobi. Is there anything that you would like to tell our female clinician engineers and future female clinician engineers? The last comment from your side.
Yeah, no, I don't have anything prepared, but for me, the field is so fun. It doesn't feel like work, and I think this is you know, I can only recommend if you love it, love what you do. Yeah, it becomes just every day is nice.
Okay. Thank you very much, Professor. So now I pass my word to my co-host, Banu Köse. She's amazing and wonderful, also an engineer, and she will present our second speaker. So, Banu, the stage is yours.
Thank you. I am Banu, a researcher on cardiovascular engineering at Istanbul Medical University, and I'll be the host of this webinar with Liana today. And at the same time, I want to say this: Marcy's presentation was amazing because it was very enjoyable to learn about her story on this. She thought that she has always worked on cartilage. So learning about what she's doing, what kind of fields she's related in, and developing successful clinical strategies on tissue engineering, repair, or bioprinting, and maybe thinking about being hopeful about artificial organs. It was very nice to listen her story and her answers to our questions. Thank you, Marcy. And now I want to tell about our second speaker, Dr. Ellen Rousch. She's an assistant professor at Institute of Medical Engineering and Science and at the same time at the Mechanical Engineering of MIT. And she's a director of Therapeutic Technology Design and Development Lab. I know her because of her inspiring research papers on cardiovascular engineering, especially about devices, about heart assist device. Her research includes development of novel devices to repair and augment cardiac function using disruptive approach such as soft robotics, combination of mechanical and maybe you adding cell therapy and use of light activated biodegradable adhesives. So she has also experience of medical device industry before her research, and she has some patents about this field. And she is the recipient of multiple science awards, including the Fulbright International Science and Technology Award, the Wellcome Trusted Award in Science, an American Heart Association Predoctoral Award, National Science Foundation Career Award, and Charles Hood Award for excellence in child health research. So today she will talk about one of her studies, and the topic of the study is integrating organic components with synthetic soft robots, a paradigm shift for preclinical testing of implantable devices. Welcome, Ellen.
Thank you. Yeah, thanks so much, Banu and Liana, for having me speak today, and I appreciate having this forum to speak to all of you clinician engineers. So I'll just share my screen and get started. So yeah, as Banu mentioned, I'm going to speak today about integrating organic and synthetic soft robots for the purpose of replicating both the anatomy and the biomechanics of organs so that we can better test implantable medical devices. So I'll start just by thanking my group up front. This is my group here at MIT. I started here as an assistant professor in September 2017. So about 3 years into having my own independent group here at MIT. And, you know, I'm so grateful for these students that make all this work happen. So this was us last summer at a lab retreat in Long Island, New York. Unfortunately, the lab retreat this year may look a little different given the current crisis, but I really appreciate working with these talented individuals, and the work I described today really is a credit to them. So just before I move into more of the research stuff, as Banu mentioned, I'm a biomedical engineer by training, and I did my undergraduate training in Ireland, in Galway, where I'm from. And then from there, I started to work in industry, both in Ireland and then in the United States. I worked in the medical device industry for about 5 years. There I worked mainly on implantable cardiac devices, including heart valves, stents, injection catheters for different types of therapies. So that was really where I, you know, became very passionate in cardiovascular implants. But while I was working in industry, I realized I also wanted to kind of go back and study more in depth some of the pathophysiology and the interaction between synthetic engineered devices and how they interact with the host, and how we can better design these devices so that they work in tandem with organic tissue, instead of replacing function, that they can assist the function of organic tissue, and potentially over time allow the tissue, the native tissue, to recover its function. So that really was the focus of my PhD. I did my PhD at Harvard with Professor David Mooney, who does a lot of tissue engineering, and Professor Connor Walsh, who does a lot of robotics. So two very different fields that I tried to meld and use to better cardiac therapies. So in my group at the moment, these are some representative images of what we do. But the overview that I usually kind of give of my research is that I work in kind of 4 different areas. One is mechanical assist devices to augment organ function. So we use these mechanical devices that we implant in the body, and they assist organs to that are diseased or that have somewhat reduced function, and these devices work with the organs. So one example is an extracardiac sleeve that I worked on during my PhD, which goes around the heart. And it contracts and twists at the same time as the heart beats, but it helps the heart to eject blood more efficiently without taking over the function of the heart. We have other work doing something similar for the diaphragm for patients that have impaired diaphragm motion and can't breathe as effectively. We are designing a device to help them move the diaphragm. That's kind of one area. The other area I'm interested in is Meccano responsive therapy delivery. So how do we get therapy, either cell therapy or drugs, to different parts of tissue that are diseased? How do we deliver it in a minimally invasive way? How do we keep it where we need it to be? And how do we refill it and replenish it and use either mechanical or light stimuli to have it be responsive? So that's another big focus area. And I'm going to talk more today about these dynamic organosynthetic simulators. So one of the main things that I realized as I was developing these devices is that it's really important to have a high fidelity test bed to test implantable devices in before you move to preclinical or animal models. It's really nice if you can recreate the environment in the lab and test your devices and iterate through different designs, you know, in a model that you trust. So I'll speak more about this, but what we do is try to use some robotic techniques and combine these synthetic materials with real tissue so that we can preserve the tissue properties and the tissue features, but we can recreate biomechanics in a controlled way as well. And finally, we use a lot of in silico models, mainly finite element models, where we can create computational models of these dynamic organs, and we can recreate their motion, their heat. For example, for the heart, we can recreate the flow inside the heart and the hemodynamics, and then we can simulate implanting devices and how this will affect the heart. So the approach I take to these 4 areas, I use soft robotic implantable devices to augment organ function, and also to modulate the immune response when we're trying to deliver therapy. And I use these same soft robotic approaches to reanimate organic tissue for better benchtop simulators. So as I mentioned, the focus today will be on these dynamic organosynthetic simulators, where we combine soft robotics with tissue. So why do we do something crazy like coupling organic tissue with soft robots? So there are limitations in the current models for testing devices. And I've shown here 4 different images. Each of these models has merit, but also has limitations. So for example, on the left, this is a biomechanical simulator where you can simulate the pressures and the flows of different chambers of the heart, which is good for testing things like valves. But obviously, it's not anatomically accurate. It doesn't look anything like a heart, and it doesn't have the same tissue properties. There are also these passive models that are used for clinician training and for kind of recreating the path to the heart, but often these are passive and made of silicone or something that also doesn't represent tissue. There are computational models, like I mentioned, that are very useful for mechanics or for fluid mechanics. But again, a lot of assumptions, and you don't get the tactile feel for what you're testing. And then there are these ex vivo models where you take ex vivo tissue and maybe connect it to a flow loop and perform your testing. That's slightly limited in that you only have a certain window of time after you extract the tissue in which before it stiffens, and obviously it's passive here unless you preserve it in some way, so you don't have the dynamic function of the heart. We're also looking at using these models for teaching and training. So these are some of the models for the lungs here. The last slide was related to cardiac models, but there are models here that are used for teaching and training. They also have limitations. So overall, I think there's a need for, there's an unmet need for dynamic, physical, tunable models that preserve anatomic features while also mimicking the mechanics and allow students or trainees to visualize and simulate different disease conditions. So I'm going to talk today about 3 different areas in which we use this approach to create organosynthetic simulators. The first I'll talk about is a biorobotic hybrid heart. So this was work that we published last year in Science, this year actually, sorry, in Science Robotics. This is Clara Park, a graduate student of mine, and this is Yiling Fan, another graduate student of mine who really did most of this work. The idea of this was that we took an explanted porcine heart, so a pig heart. Pigs have a similar size and structure to human hearts, so it's good for testing cardiac devices. And we used a technique that has been reported before in the literature for unraveling the left ventricle, or the entire ventricles of the heart. So something called a helical ventricular myocardial band. And actually what you can do is essentially unwrap the whole heart into one band. It's really fascinating. So then we could take a part of that corresponded to the left ventricle, which is the pumping chamber that pumps the blood around the body. And then we collaborated with the Professor Chris Nguyen at MGH, or Mass General Hospital. He's also part of the Martinos Imaging Center, and he does advanced cardiac magnetic resonance imaging, particular, we use diffusion tensor imaging, which you see here. You can basically use a technique called tractography to outline the orientation of the muscle fibers in the band. So now we have this 2D band and we scan it, and we're able to identify the orientation of the muscle fibers in the band. Then we use soft robotic techniques to replicate that, and we orient these actuators, which are essentially small bladders embedded in a silicone matrix that we can pressurize, and that will contract in a predetermined fashion. So we have this soft robotic synthetic myocardial band. Meanwhile, we take away the real tissue from the ventricle, and when we rewrap around the dissected ventricle with this soft robotic band, we can integrate everything into one component. So we have organic tissue, apart from the myocardium of the left ventricle, or the muscle of the left ventricle, which we now replace with a synthetic soft robotic muscular structure that we can control and we can move, and which should move in the same way as the real ventricle. So to do that, we had to kind of come up with a lot of different processes to enable us to achieve our end goal. So we took a pig heart ex vivo. It's like explanted from the body after the animal was used for another procedure, and we can fix it. And there's a lot of different ways we can fix it that will preserve some of the tissue properties, but enable us to use the tissue for much longer without it expiring or getting contaminated. And what we can do then is we embed the atria, or the upper chambers of the heart, in the vessels, we just encase them basically to protect them while we're working on the ventricles at the bottom. And then we can use a cast to cast the shape of the lower ventricles. So we have the outer surface of the heart replicated. Then we go through this dissection procedure, which this is a very careful layer-wise dissection of the explanted heart. And you basically bluntly dissect in a manner that follows the cardiomyocyte, or the muscle fiber orientation, which allows you to dissect it in a very predetermined manner. So you're left with this left ventricle that has no muscular tissue surrounding it anymore. And the first thing we wanted to do was see if we could replace that muscular tissue with just silicone, just passive silicone, without any actuators or robotic components. So we placed the dissected heart back in the outer mold we made in the first place, and we filled the gap that had been dissected with silicone. So you essentially recreate the ventricles that you dissected with the silicone structure. Then we had to come up with an adhesive that would work, and this was no easy task, to come up with an adhesive that would adhere fixed tissue to silicone. So we worked with collaborator Professor Zhuanhe Zhao, who's at MIT in the Department of Mechanical Engineering. And we came up with an adhesive that worked well for this purpose. So we coated the dissected ventricles with the adhesive, and then put these two parts together. So what we had is this passive biohybrid heart, but as you can see, it looks pretty much like one piece, and we achieved really nice coupling between the synthetic structures and the organic structures. This here on the top is SEM images of the adhesive and how it interpenetrates into the tissue, which forms a really nice bond between the tissue and the adhesive, which then can bond to the silicone. So the green there is the tissue sil, as we called it, the adhesive, and this brown layer is the cardiac tissue. Here's a CT image where the red is the heart tissue, and in yellow you can see the tissue sil adhesive. The gray here is the silicone matrix. So again, you can see this is a cross section A here, and B, the left ventricle is here and the right ventricle is more crescent shaped, but you can see how the adhesive conforms really nicely between the silicone and the tissue. But as I mentioned, we wanted to make this active, and just to go through this myocardial ventricular helical band a little bit more. This is the heart, and this is, as you start to unravel it, what it looks like. So you do this in a very systematic fashion. And then you end up with this band, and you can identify the parts that form the left ventricle, which is the active part we wanted to. So we just isolated those parts that form the left ventricle, and we went to the scanner. And this is diffusion tensor MRI. So basically, you can quantify the direction of fastest diffusion of fluid in the tissue, and that corresponds to the orientation of the cardiac muscle. So we did that, and then we could get this 3D structure of this flat left ventricle, essentially. And then we projected that data onto a 2D plane to allow us a 2D structure where we could perform tractography, which simplifies it and enables you to identify the direction. From that, we came up with a coarse grid with the orientation of the fibers. And then we transferred that into a CAD layout of a soft robotic band. You can see here, obviously, we don't get anything close to the resolution of the cardiac fibers themselves, and that's an ongoing work where we're trying to get higher resolution soft actuators into the band. But what we show here is they're called flat pleated pneumatic artificial muscles, and they're made from 2 layers of silicone, and sandwiched in between the silicone layers you have paper structures that are inextensible. And then in between the paper layers you have PVA, which is a dissolvable structure, so you're left with a bladder basically in between the paper and the silicone layers. And when you pressurize that, as you can see here, almost kind of like bubble wrap, you get radial expansion of these little bubbles, but also linear contraction. And we can create finite element models to predict the stresses in those actuators, and also the strain, what kind of strain we'll see when we pressurize them. And we can characterize experimentally these actuators. The reason we went with these flat versions for this is that because we needed to wrap it and fit it back in the cardiac geometry, we didn't have a whole lot of real estate to place bulkier soft actuators. So this video just shows briefly the structural image. This is using MRI again. The soft robotic part is in dark gray and the tissue is in light gray. And as you go through from the front to the back, you can see the different layers and how the 2 materials conform well together. This is, if you imagine, coming from the top of the heart down. Left ventricle, right ventricle. Again, the light is the tissue and the darker is the soft robotic matrix. You can see some of the tubes there at the end, which we use to pressurize the soft actuators. Then we show the motion here. So when we pressurize these, we get motion of the left ventricle. So this shows the left ventricle moving under echo and from a cross-sectional view. And this is reference of an actual heart. So you can see from end diastolic to end systolic, which is the end of the relaxation period of the heart to the end of the contraction period of the heart, you get this contraction of the ventricle. And if I play that again, we see similar contraction of the ventricle in the biorobotic hybrid heart. And we saw similar percentages of contraction with the robotic heart compared to the reference heart. We still have some ways to go to fully recreate the hemodynamics or the pressure volume loop of the heart on each cycle, and that's the subject of ongoing work in the lab at the moment. So yeah, we've successfully coupled synthetic active myocardium with preserved organic tissue, and we were able to recreate myocardial motion. The next steps are to recreate the PV loop and attach to a mock circulatory loop so that we can have a full operating mock circulatory loop, which will allow us to test implantable cardiac devices, mainly like valves and occluders for that are, intracardiac devices that are placed in the heart. These are subject to the 3D motion of the heart, and they also change the hemodynamics. So, we really want to test these in this type of setup. We'd also like to decellularize the tissue. So right now we're fixing it, but ultimately we'd like to fully decellularize it before combining it with the soft robotic matrix. And then we could think about using it as a total artificial heart. So we're moving towards that as a long-term goal. And I'm lucky to be part of a Hybrid Heart Consortium, which we're currently finalists for the British Heart Foundation Big V Challenge. So we're working on a submission for the final round of that funding. The second thing I'll talk about is an organosynthetic model of respiratory mechanics. So here we're trying to recreate the respiratory system. What we are doing this for, this is the vision, is for a number of reasons. One, as a teaching tool for students and patients and the public to visualize what happens during breathing. So I teach respiratory pathophysiology at Harvard Medical School, and I teach more the engineering side of it. So we've brought this simulator in to teach the students, and I think it's an effective tool to help visualize what's going on and measure the pressures in the different compartments. We also can use it as a training model for ventilators, for mechanical ventilators, to test their function and to test the changes that happen in the pressures in the thoracic and the abdominal cavity when you attach a ventilator to a respiratory system. And we can also, again, use it as a test bed for testing devices that are used in the thorax, or for different surgical strategies that are used for respiratory pathophysiologies. So very briefly, we have a sealed acrylic box and acts as the thoracic cavity, another one that acts as the abdominal cavity. And we have a synthetic diaphragm that is actuated by these black artificial muscles. Again, they're pneumatic, so when you pressurize them, they'll contract downwards. So they'll pull the diaphragm down, which happens during inspiration. The diaphragm moves down, increasing the volume of the thoracic cavity and decreasing the pleural pressure, so the lungs will inflate. And we have attached organic lungs into this setup, and we've 3D printed an anthropomorphic rib cage, which goes in the thoracic cavity as well. I should mention that this is Marcus Horvath and Lucy Hugh, two senior grad students in my group that have done excellent work on this simulator. We published this recently in APL Bioengineering as well, if people want more detail on it. But this is what it looks like when it's realized. And these are the pneumatic artificial muscles connected to airlines. So when we pressurize them, this whole diaphragm will move and thus inflate and deflate the lungs. We have pressure sensor here and here, and we have a spirometer or a flow meter on the trachea, which will allow us to measure pressures, flows, and volumes. And what we can do is vary the pressure input that we give to these pneumatic artificial muscles, as you see here. And that has an effect on varying the change in the pleural and the abdominal pressure that's seen in those cavities. So we can tune the amount, like shallow or deep breathing, we can tune the amount by which the diaphragm moves. This video will show the motion of the simulator. So here it is in the lab. You'll also notice we have a compliance window for the abdomen that we can change. So as the diaphragm moves, these lungs will inflate and deflate. And I won't go into too much detail here, but what we can also simulate is pathological conditions. So we can change the compliance of different components, like both the lungs, we can change the pleural space, the contents of the pleural space, and we can change the abdominal compliance to simulate different disease conditions, because these all have effects on breathing, obviously, and on how the pleural and abdominal pressures change with varying degrees of diaphragmatic motion. One example of what we're using this for is for testing of devices. So I teach a medical device design class to mechanical engineering students, and one of the student teams came up with a tool that's used for placing a patch on the lungs when there's been a laceration or a puncture to the lung, and this patch seals over the puncture. So we could use our setup to test this and to measure breathing function before puncture, after puncture, and then after closure. So we could have a standardized way to measure if the patch was sealing the lung, and also how easy it was to deliver through the rib cage. So we could measure the pleural pressure over different breathing cycles before and after sealing, and how the tidal volume changed as a function of baseline before or after puncture and then after sealing again. Also of interest at the moment is ventilators, and we could use this test setup here to measure ventilator function. So the simulator had very similar measurements, both in terms of volume and pressure, to what the ventilator was measuring. And what we also showed is that we can use the simulator to trigger a breath from the ventilator. So if we, like if the patient starts to inhale or exhale, the ventilator will trigger that part of the breathing cycle, and we could use our simulator to do that. So to summarize on this part of the talk, we were able to replicate breathing mechanics, pressures, volumes, and flows. And the next steps are, we want to evaluate this respiratory simulator setup with bronchoscopy. So doing, a scope into the bronchi, and we could do that during breathing. So we see what the doctors see when they're doing this in a patient. We could also use it to test stent placement and cryoablation. These are different types of procedures that are done in the lungs. We also, and I'll talk a little bit more about this in the next section, we want to couple this respiratory simulator with a mock circulatory loop. And this is to assess and demonstrate the interdependence of the heart and the lungs. So the cardiovascular and the respiratory systems are very interdependent, and if we can show that and replicate them both together, it's very beneficial. This is something that doesn't really exist at the moment. And then finally, we have an ongoing project, like I mentioned, in the mechanical assist subgroup of the lab, where we're looking at assisting the diaphragm with an implantable ventilator. So a device that goes in to move the diaphragm for patients that have muscular dystrophy or diaphragm paralysis that can't move their own diaphragm. So this would replace a tracheostomy where the patient is ventilated through here. And this simulator will allow us to assess the function of that device before we move to animal studies. Finally, I'm just going to briefly touch on a project where we're combining this respiratory simulator with a cardiovascular flow loop. This is for what's called the Fontan Physiology. I'll describe that a little bit more. This is a collaborative project with Dr. Pedro Del Nido, who's the Chief of Pediatric Cardiac Surgery at Boston Children's Hospital. And these are, Marcus is a grad student of mine, and David is a visiting student actually from ETH, who has been working on this in the lab for the last close to a year now. So this is a normal heart. There's two ventricles, two atria, but often, well, not often, but in some cases, kids are born with this condition called hypoplastic left heart syndrome, which is, a congenital cardiac anomaly where these kids have a very small or hypoplastic left ventricle, and the left ventricle pumps the blood to the body. So if it's underdeveloped, it's a big issue for kids. And what happens is they undergo surgery. They undergo about 3 surgeries by the time they're 5 years old. The final one is usually called the Fontan Surgery. And basically what it does, this is a normal cardiac system. This is a single ventricle after Fontan. So what happens is the one functioning ventricle is replumbed basically to pump the oxygenated blood to the body. And then the surgeons put in a shunt, which means that the return to the lungs is passive. So there's no active pump to return the blood to the lungs. And we're working on a device to improve this and to make an active pump to return the blood to the lungs. But before we do that, we need to recreate this physiology in a simulator. This just shows the different cardiac surgeries that these kids go through and at the different ages. So this is a kid, Judah, who his parents have kind of shared his story online and his different surgeries that he's gone through. So the first is a Norwood procedure, then they insert what's called a bidirectional Glenn, and the final is the Fontan. I won't go into too much detail of the cardiac surgery there, but the result is that this right ventricle is now becomes the systemic pump to pump oxygenated blood around the body. So one of the big things that's very apparent in these patients is that the respiratory system has a big effect on the venous return of blood to the lungs, and the mechanics of the respiratory system are big influencers of that return. So here we have combined the respiratory simulator with an anatomically accurate model of the reconstructed or the Fontan physiology after the last stage of that surgery. This is called a Fontan cross. And what we can do is connect that then to a mock circulatory loop. So this pink fluid here is a blood analog, and then we have different pressure chambers where we can vary the pressure and vary the height to represent different parts of the body outside of this thoracic cavity. So by flowing blood through here and then by moving the diaphragm and replicating thoracic pressures, we can see the effect that those pressure changes have on the blood return. So this shows in action with the diaphragm moving, and you can see here in the there's air in this system, but it helps to visualize. There typically wouldn't be air, but you can see as the diaphragm moves, this is with no flow going through it at all. It has an influence on the flow in the superior and inferior vena cava, and that's been really helpful for us to test out different strategies for helping with these patients, because we can now see how breathing affects it. So we're collaborating with these clinicians to try to validate this model with imaging of patients, so that we can validate that this model is accurate, and then potentially it can lead to new insights and new treatment modalities. There was a recent paper that described how the IVC, that's the inferior vena cava, how the flow in the inferior vena cava changes with the respiratory cycle, and we were able to recreate that with our mock loop. So what you see is at a certain point there's actually retrograde or backward flow in the inferior vena cava, and that's what we're trying to circumvent and to prevent with our device. So to conclude, we replicated the Fontan hemodynamics, especially this interdependence with respiration. We're now moving to validate with patient MRI and to use this as a test bed to evaluate implantable devices to improve the quality of life for these patients. I'll finish with an exhibit that we had of these organosynthetic components, the biohybrid heart and the respiratory simulator. This was in the Science Gallery in Dublin in Trinity College. Marcus Horvath and Luca Rosalia put this simulator together. I'll just show a short clip of it operating, but it was on display, an active display where people go into the exhibit, could press a button and the diaphragm would move. So here we see the diaphragm moving and the lungs expanding. And Marcus went to introduce it to people and describe how these muscles work. This is the heart, which isn't active at the moment, but you can see it in between the lungs and the lungs expanding as the diaphragm moves down. Okay, I am going to leave it there, and I want to thank all my funding sources. Obviously, without funding, this work wouldn't happen either. And again, just thank my fantastic research group. So thank you, and I'm happy to answer any questions you might have.
Thank you, Ellen, for this mind-opening and motivating speech. So, as being one in cardiovascular engineering field, I was very excited when I'm listening your presentation. And I have lots of questions. Actually, most of time in congresses or seminars, we are looking for small, short coffee breaks to ask questions. But now we are here and I can ask my questions easily. So I want to ask about that heterotoxic patients, just Fontan patients. Actually, I am working something on some kind of thing, hypoplastic left heart syndrome. Mostly, you are working on just mimicking. Just you are using MechE systems for mimicking the physiology of the pathophysiology. So when you are you told that you try to mimic the post-operated physiology like with shunts or grafts. And how do you obtain that model? Are you using MRI image slices to obtain the last version of the geometry, or you are implanting that graft on your mock model that you have prepared before?
So I'm sorry, the question is how do we get the geometry for the active graft that we're designing for the Fontan Physiology? Is that?
Yeah, actually, you told that you are trying to understand the mechanism of the post-op geometry, post-op flow, and post-op circulation. So do you obtain these models from MRI or CT, or you also have created something idealized and you are adding that kind of graft in the same diameter, for example, to your mock system?
Yeah, no, I totally understand. Yes, we do. We get the geometry from patient MRIs or CT scans. We reconstruct it in a software, and then we 3D print the geometry. At the moment, we have one from a patient that we're using, but this is highly heterogeneous, so we could print and replace. It's pretty modular, so we could replace that component with different patient-specific anatomy pretty easily.
Okay, I understand. And what are the materials are you printing from? Actually, it is, you told at the first, there are some models, but not like tissue, but what kind of resin materials, silicone molds, or some other kind of composite materials for printing?
Yeah. Depending what kind of interventions we're evaluating, we choose what material we print from. So if we're not implanting a device in that location, potentially replicating the tissue properties like the internal surface friction isn't as necessary as it may be for other devices like intracardiac devices. But we do a number of different types of material. We print in flexible materials, but then we also print molds, which we can then use to cast different geometers of silicone or urethane to allow us to kind of have tunable properties in the vessels. And we're also interested in creating models of vessels that have variant compliance. So if we have a bladder or an actuator on the vessel itself, we can use vacuum or use a type of laminar jamming actuator to change the properties, because the compliance of vessels changes over their geometry and over with different conditions. So if we can replicate that, that's advantageous as well.
Yeah, it is not easy to find the consistency just between physiological data and your mock system. So it is not easy, because you also showed a mechanical simulator, just pulse duplicator kind of something in the first slide of you. So it is not easy to find the same results at the same time, but just you are just using approximation, some other kind of methods like finite element models, prototyping, and mechanical simulators, you obtain just very nice stuff, so you can turn them into products. That's very nice, because it's the same process, just like finding something and trying to use it for the beneficial of the health science. So you have lots of patents, I know, because of that. Because you came from industry, so you first worked there in medical industry, so medical device industry. When you are doing your research, do you plan the patent process or ISO certifications or CE, FDA mark processes at the same time simultaneously, or after research you combine all stuff to find the university industry collaboration?
Yeah, that's a good question. So I think probably the IP and regulatory considerations are a little bit separate. For IP, we would try to, as Marcy mentioned, try to file before we publish, and before we submit a manuscript, we'll talk to our technology licensing office and see if there's interest in filing a provisional patent, or before we present it at a conference, for example. In terms of regulatory approval, I think that comes after the research and development stage in the lab. Most of the devices we work on in the lab are pretty early stage, so we're coming up with concepts and trying them out in these models or then in animal models, and we're trying to figure out technology. So it's not at the point where it would be a device that could be approved for human use at all. What we've done in the past, and we patented, and if there are companies that are interested in licensing it, we can work with the company and then take it through the kind of translational path and then get regulatory approval.
In the future, especially in the field of cardiovascular engineering, which topics should be emphasized for? You are working with different parts of this field, like soft robots and mechanics and some surgical methods and respiratory system. So what do you think for the future studies, what you are planning to work on?
Yeah, I'm interested in all those areas. I think, the technology is changing very quickly in this space. So, things like stents and prosthetic valves have become, very commonplace in the last couple of decades, and it's been an amazing change. And I think now, people are looking at, can we make them degradable, or can we make them tissue engineered valves, or bioprostheses? So moving towards less permanent metallic objects and more kind of biomimetic approaches. I think people are moving as well away from either a surgical or an interventional approach, and maybe some sort of hybrid of both.
Yeah.
But things are getting smaller and less invasive all the time, which is positive. I think obviously monitoring and sensing are getting a lot better, so we have more data of when patients might need interventions and how interventions are performing, how patients are doing afterwards. And for things like left ventricular assist devices, I think it'll move towards smart devices that can sense and kind of adapt with the patient. So yeah, I think, more biomimetic devices, more sensing, more smart devices is kind of the way things will go.
Yeah, thank you. But at the same time, you have a background of laparoscopic devices at the same time. So actually, the idea is the same. There's a problem, and understanding the mechanism and finding a solution due to the problem. So can you tell about that laparoscopic research you did before? Because of your medical device background,
yeah.
I want to learn, can we you told about the methods you are using, and you also have some methods patents. So you are using some methods, some tools. Because lots of students are listening us, so they want to understand and they want to find solutions related to engineering and clinical background of them.
Yeah.
So maybe telling about just combining these methods to other something related but different fields. So maybe you can briefly tell us about this, or something you do related to these things using the same methods.
Yeah, I mean, I think my industry background was more in interventional, cardiac endovascular devices. I think we still use those techniques. So, designing devices that will be able to fit into a catheter and can be navigated to a remote part of the body internally and then deployed there under image guidance, which avoids a very invasive surgery. We still use those techniques all the time, both for we're trying to develop a minimally invasive device to deploy our heart sleeve, for example. We also use minimally invasive ways to deliver reservoirs to deliver therapy for localized delivery of bioagents. So I think those methods are very important for us. And how do we make things in a way that we can deliver them without being too invasive? And I think one of the key things, and it's probably relevant for this webinar series, is that it's important for engineers designing these devices to understand the pathology and really understand how this device is going to work when it's implanted, and the interaction between the host and the device, the effect of the device on the host, but also the effect of the host motion or immune response on the device. So it takes good mechanical engineers, but also, it takes a lot of in-depth understanding of the physiology and the pathology and the biomechanics.
There's a question from Lady Catherine Meja Perez. She says, thank you so much for such a fantastic talk. So many individuals would benefit from these developments. Can you tell us about how to jump from the development process to application in clinical scenarios? Which entities need to be involved?
Sure, yeah. So in the lab, we're really focusing on that early R&D stage. And to get this into the clinic, we obviously need to work with the surgeons or the interventionalists that are going to be using it to make sure that there's a need and that it's easy to use. And then usually we would license the technology, or potentially have our own startup. So we need help there on the licensing and business development side. We'd also need then to go through more preclinical studies and clinical studies, and we need to get approval from regulatory bodies. So that brings in teams of preclinical people that can plan clinical studies, regulatory affairs people. So a lot of kind of multifunctional teams outside of the research and development team are needed to actually get this to the clinic.
Yeah, thank you. You have experience with working at the same time clinicians and engineers in harmony. So what is missing or what can be improved in the process of producing solutions in harmony with each other as clinicians and engineers? What qualifications would you suggest that students now listen to us and feel excited to do something in this interdisciplinary field?
Yeah, for me, it's really exciting working with clinicians and coming up with these devices. And I think it's more communication earlier, more often, just really, it should be co-developed and co-invented, because, people have very different backgrounds and see things from different ways, but it's important that it's not designed just by an engineer, that also the users will have input into it from the beginning. So a lot of the devices that I've worked on that have been more successful are the ones that I've worked with surgeons or clinicians from the beginning on.
Yeah, it's very important to have that connection and understanding using the same language maybe, because you are teaching medical device design or optimizing design, and at the same time you teach pathophysiology, or something on pathophysiology. So understanding and finding the common points or having the same backgrounds in some topics would be better to understand each other. So I think your students are, the people in your lab are very lucky being with you and learning from your experience.
Yeah, I mean, I think the graduate program in the Institute for Medical Engineering Science is called the Health Science and Technology program, and there's kind of co-education of medical students with engineering PhDs, and the engineers will take rotations, clinical rotations. So it's really immersion in the clinical setting, and I think that really helps.
Yeah, that really helps. It's fantastic. Thank you, Ellen, for being with us today. It was very inspiring, and learning from that innovative technologies and development, the last developments in this field was fantastic. Thank you very much.
Thank you for having me.
Thank you. Thanks for everyone listening us, and thanks to our amazing speakers. And actually, I want to thank for the founders of Clinician Engineer Hub, Dr. Neil Sharma, Dr. Ali Yetishan, and Dr. Matt Spargot for these unique and fruitful meetings. They have organized these meetings, and I and Liana were the hosts. So this July, there will be 2 more webinars on Saturdays. For just there will be speakers of, again, female engineers for this July. They will share their success stories in different fields. So just engineering, but relevance to healthcare. And we will be, I and Liana will be the hosts of these webinars. So thank you for your time, and I wanna, Liana will give you a closing speech now. I think maybe she will tell about our next webinars and next speakers. Thank you, everyone.
Thank you again to our amazing speakers from today. Yes, as for the next week, we have another amazing webinar with other amazing female engineers. I will just share my screen very quickly. As you can see, we will have Dr. Sara Gohar-Sizadeh from University College London. We will also have Ifeyama Rita Kanu, founder and director of Intelligent Limited, and of course, Pia Situ, who is the chair Industry Advisory Board at UNSD Computer Science and Engineering. So please register in Eventbrite and join us next Saturday. As for today, we can close the session, but please, everyone, turn your cameras on so that we can take a last group picture. We have Lady Catherine and all the participants. We welcome everyone to put their cameras on so that we can have the picture. I just want to thank both Clinician Engineer Hub and, oh, someone is trying, but it's not working. Let's see. Yes. I want to thank Clinician Engineer Hub and EIT Health alumni for this great opportunity to present our amazing speakers in this webinar series as well. And we are just waiting for a couple other participants to put their cameras on. Hello, Roa. Hello, Miriam. And thanks for staying with us until the end, all of you. Okay, I will then take the picture. We have some people that are shy. Okay. It's done. So thank you everyone for your time, for your availability, for our speakers for your great speeches and very motivating speeches as well for our clinician engineers. And we will see you next week. Thank you very much.
Thank you.